Okay, so active transport, we should see ATP coming in and helping out. So we've got to have an energy. The reason you have to have energy for these, and this is occurring, a sodium-potassium pump, this is occurring when we're doing muscle contractions, nerve impulses. So we like to keep, say, potassium on one side, and we like to keep sodium on another side. Thing is, with ATP, why it's needed is because we're going against the concentration gradient. Now we're pushing stuff out or taking stuff in that doesn't want to be there because there's already a bunch of them in there. So if you're looking at potassium, we've got, first we'll look at sodium. Sodium's coming in and we're shooting it out. Doesn't really want to be there because there's already a bunch of sodiums there. Same thing with potassium coming in and we're shooting it inside the cell. It doesn't really want to be there. There's already a bunch of potassium particles. So we're having to push it. And that's why you need the energy of ATP to actually do that. Maybe this one will load. Come on. So it's going against the concentration gradient. That's why we need ATP.
In active transport, solutes move up the concentration gradient from areas of lesser to greater concentration. One of the most important examples of active transport in the body is the sodium-potassium pump. The sodium-potassium pump works to transfer sodium from inside the cell, where sodium levels are low, to outside the cell, where sodium levels are higher. Whereas transferring potassium from the extracellular fluid, where potassium levels are low, to the cell's interior, where potassium levels are higher. The process begins as 3 sodium ions from inside the cell funnel into receptor sites on a channel protein. Fueled by ATP, the channel protein releases the sodium ions into the extracellular fluid, causing them to move from an area of lower to higher concentration. Meanwhile, 2 potassium ions from outside the cell enter the same channel protein. The potassium ions are then released inside the cell. This keeps the concentration of potassium higher and the concentration of sodium lower within the cell. Just like swimming upstream, moving against the concentration gradient requires energy, which is provided in the form of ATP. The sodium-potassium pump is responsible for regulating the volume of fluid within cells, providing the electrical potential necessary for nervous system activity, and helping in heat production.
I like that example of swimming up the stream. So if you're going with the current and you're swimming downstream, you can just float, and it would carry you downstream. That's all passive transport. Whereas active, you would be turning around and having to swim against the current. That would take a lot of energy or a lot of work to do. Okay, so this is talking about transport inside the cells where we have vesicles. When you were looking at endoplasmic reticulum, transporting proteins from the Golgi body to the outside of the cell, they did little balls and spitting it out. That's what we're talking about. So endocytosis, N like IN, is stuff going to be going out of the cell or in the cell? It's going to be taking stuff in the cell. Exocytosis, like exit, it's going to be going out of the cell. And I love my first video that I found on exocytosis or endocytosis, taking it in the cell. So endocytosis. This would be a white blood cell.
Like that.
So this is one of the types of endocytosis. There's phagocytosis and pinocytosis.
Phagocytosis, the process by which certain cells alter toxins and other organic material by wrapping their cell membrane around it and incorporating it into the phagocytic cell, where enzymes break down the chemicals and convert them to useful nutrients.
Huh?
Well, perhaps I can demonstrate. Watch carefully, boys and girls.
Very simple, endocytosis. So, phagocytosis, with this type of endocytosis, phagocytosis happens quite a bit. Oh, please load. So if you have a bacteria and your white blood cell is rolling along and sees it, it can engulf it and take it inside itself. And actually the lysozyme will digest it and break it down. So that's the great thing about your endocytosis. Penocytosis, which I don't have a video on, penocytosis is taking in something, but it's not material, it's water. It's taking in fluid inside the cell itself. So it's doing the same thing as going around and engulfing something and bringing it inside itself, but it's water this time.
This is like totally like awesome, but it's about the worksheets. Does connective tissue have like a good blood supply or?
We'll talk about that'll be next tomorrow's lecture. Okay, so exocytosis, going outside or exiting the cell. This one doesn't have any music or anything to it. So hopefully you can see with the lights on. So this here you should see, and they'll move it up, they'll move the microscope up so you can actually see. It's on a high level buffer there. But it's going to show with exocytosis, materials exiting the cell via vesicle, if it ever loads. Come on.
I hate that word.
Yeah, it's not going to load. But I'll have these online as well, so you'll be able to view them. Our internet must be having a field day. Okay. Let's look at DNA occurring there or being produced and transcribed inside that nucleus. You do want to know how these match up. So how our DNA matches up, T and A, C and G, and as well as how they have to be transcribed. So when our RNA, our messenger RNA comes in and transcribes. So there is another way that that order will change, and we'll look at that in just a minute. But you do need to know this for the test.
The DNA, deoxyribonucleic acid molecule, is one of the largest and most complex of all molecules. It is a polymer made up of many smaller molecules joined together in a sequence. The building blocks of DNA are millions of pairs of nucleotides. Each nucleotide consists of one sugar, one phosphate group, and one of 4 possible types of nitrogenous bases. The 4 types of bases are adenine, A, thymine, T, guanine, G, and cytosine, C. The phosphate group alternates with the sugar deoxyribose to form the backbone of the latter. 2 different bases join together to form the rungs of the ladder. Base combinations follow a specific pattern. Adenine can only pair with thymine. Guanine can only pair with cytosine. The resulting ladder is twisted in the form of a double helix. A single strand of DNA would stretch about 2 inches if uncoiled. DNA is coiled to fit inside a microscopic nucleus. The sequence of bases is the genetic code. This code provides all the necessary information for building and maintaining an organism. Essentially, DNA serves as a cell's blueprint for life.
So T tells each and every cell what it needs to be doing, where it needs to be, and what it needs to do, what its function is. So your DNA, we said we have to know how it matches up. T and A, C and G, T and A, C and G. But when we have RNA, because remember, DNA is inside the nucleus. We said it's about 2 inches long, which is really, really long. It's one of the largest molecules. So we can't get outside of that nucleus. So to be able to get that information, that code from out of here to the cell itself inside the cytoplasm to tell it what to do, what to grow into, then we have to have RNA, which will go inside there and it will transcribe that DNA code. So it'll copy itself on it, and then it can leave the nucleus and go out and tell, well, this is what DNA said you need to do. So the difference with it is we do not have a T with RNA. So you'll have a U instead. So it will match up. So mRNA, your messenger RNA. C and your G and C will still match up. We don't have a T on RNA, so it's going to be a U instead. That's the one thing that will change, is you will have a U to an A instead of a T to an A, like DNA. Why in the world it had to be difficult, I don't know. G and C, A and T, we can do, because look, DNA, and the red is our RNA. We do have an A. We do have adenine. So C to G, A to T works, C to G. Over here on RNA, we do not have a T to match up with A, so we have to use uracil. So you do need to know that little bit, the transcription. Know that DNA is too big to come out of the nucleus, and know that RNA is the one that will go in and copy its strand down. Now, in the cytoplasm, we said that we have, I'm going to go ahead and break my videos.